Okay, let's go to the next topic about how generation of cytosolic acetyl CoA. In physiological and normoxic conditions, glycolysis or beta-oxidation-derived mitochondrial acetyl CoA represents the major source of cytosolic acetyl CoA upon transpiration. That said, there are at least two relatively ubiquitous metabolic circuitries through which cells actually produce acetyl CoA in the cytosol. First, cytosolic acetyl CoA can originate from glutamine reductive carboxylation, especially when glycolysis is blocked in hypoxic conditions or in the presence of mitochondrial defects. Upon uptake from the extracellular milieu, which is mediated by various transporters depending on cell type, cytosolic glutamine is metabolized by glutaminase GRS to generate glutamate, which enters mitochondria through the age-dependent glutamate asparated transporter SRC25A13. Mitochondrial glutamate is converted into a ketoglutarate reaction catalyzed by glutamate dehydrogenase 2, GLUT2, which undergoes reductive carboxylation within the TCA cycle to generate citrate. Finally, citrate can be exported back to the cytosol where the decarboxylate antiporter solute carrier family 25, that is called SRC25A1, and converted into oxaloacetate and acetyl CoA by ATP citrate-lyase ACLY. Second, a cytolytic counterpart of ACC1, for example, acetyl CoA synthesis chain family member 2, that is called ACC2, employs acetate to produce acetyl CoA in an ATP-dependent manner. Cytosolic acetate can derive from the extracellular milieu upon uptake by various members of the monocarboxylate transporter protein family, or it can be synthesized from ethanol-derived acetylhalide by a cytosolic variant of ALDH2, namely aldehyde dehydrogenase 1, ALDH1A1, at least in hepatitis. Although circulating isolate levels are relatively low in modern humans as compared to their ancestors owing to dietary changes, the ACSI2-dependent conversion of acetate into acetyl CoA has been shown to be predominant in primary and metastatic malignant cells of various origins, especially in hepatic conditions. So, the ectopic acetyl CoA generation, as malignant cells process through the S phase of the cell cycle, functional PDC can translocate it from mitochondria to the nucleus, catalyzing the ectopic synthesis of acetyl CoA from pyruvate. Interestingly, two other acetyl CoA generating them, namely ACLY and ACSI2, have been localized to the nucleus and linked to cell growth and proliferation. So, what's the metabolic fate of the mitochondrial acetyl CoA? Normally, mitochondrial acetyl CoA is metabolized within the TCA to yield NADH, the main substrate for ATP synthesis via oxidative phosphorylation. Some cells, however, including hepatocytes, can employ mitochondrial acetyl CoA to synthesize ketone bodies. This metabolic pathway, which is catalyzed by the sequential activity of the HMGCS2, HMGCL, and BDH1, is particularly active in conditions of fasting or caloric restriction, when the majority of mitochondrial acetyl CoA derives from beta oxidation. Liver-derived ketone bodies enter the circulation and are readily taken up by neurons and cardiomyocytes, which employ them to produce ATP upon reconversion to acetyl CoA and entry in the TCA. So, what's the metabolic fate of cytosolic acetyl CoA? Cytosolic acetyl CoA is a precursor of multiple anabolic reactions that analyze the synthesis of fatty acid and steroids, as well as specific amino acids, including glutamate, proline, and arginine. The rate-limiting step of lipogenesis is catalyzed by ACAC, that is a biotin-dependent enzyme that irreversibly carbonates acetyl CoA into malonyl-CoA. In energy-low conditions, that means that when the cellular is in low ATP, high cyclic AMP levels, ACAC is inhibited upon phosphorylation by protein kinase AMP activated. In line with this notion, the cytosolic pull of acetyl CoA increases upon AMPK activation, at least in yeast. In most hepatocytes, the knockout of both ACAC and ACACB causes the hyperacetylation of extramitochondrial proteins, but the hypoacetylation of mitochondrial proteins. That suggests that the utilization of acetyl CoA for lipogenesis has a significant impact on acetylation reactions. Spermidine also consumes cytosolic acetyl CoA as it acetylates polyamines to facilitate their creation from cells and in fine their excretion with urine. Cytosolic acetyl CoA is required for cell type specific metabolic circuitries such as synthesis of acetylcholine, implying that the survival and activity of cholinergic neurons are particularly dependent on acetyl CoA. So, the compartmentalization of acetyl CoA metabolism. Acetyl CoA exists in separate mitochondrial, periaxomal, nucleus-cytosolic, and interarticular pods. Indeed, the inner mitochondrial, periaxomal, and reticular membranes are impermeable to the highly charged acetyl CoA molecule. Conversely, nuclear pods allow acetyl CoA to freely distribute between the cytosol and the nucleus. Of note, the endoplasmic reticulum doesn't contain any acetyl CoA generating enzymes, implying that the acetylation of proteins within the ER lumen critically depend on the import of acetyl CoA from the cytosol. Taken together, these observations suggest that the bioenergetic and signaling functions of acetyl CoA are finely regulated by compartmentalization and interorganular acetyl CoA flux, which are influenced by the subcellular localization of acetyl CoA generating reactions. Okay, that's all. Thank you.